SCID, severe combined immunodeficiency. In 2007, I took care of a 2-year-old patient with SCID when I worked as a nurse in Thailand. That boy and his mom had been admitted in the hospital and receiving challenging treatment for almost his whole life, around 8 years. He was extremely susceptible to any infection. He received gene therapy treatment and also chemotherapy, but eventually he defeated after fighting for 8 years. Moreover, the medical expenses was written off for his single mom. SCID is very rare genetic disorder that causes life-threatening problems. It is a type of primary immune deficiency which are characterized by abnormalities with responses of both T cells and B cells. About 1 in 58,000 babies are born with SCID in the US each year. SCID may be caused by mutations in any of several genes and can be inherited in an X-linked recessive disease most commonly, or autosomal recessive manner, which are due to mutation in disease-causing genes on other chromosomes. The best known form of autosomal recessive SCID is caused by adenylogenease AGA deficiency, in which infants lack the AGA enzyme necessary for T-cell survival. Boys with this type of SCID have white blood cells that grow and develop abnormally. As a consequence, they have low number of T cells. They also have low numbers of natural killer cells, and their B cells do not function. From cell biology class, I've learned and gained more insight about cellular expressions that can explain the pathology of the disease. For example, cell signaling that produce the components of T and B cells in the body. For cellular expression, IL-2RG interleukin-2 receptor subunit, the protein encoded by these genes, is an important signaling component of many interleukin receptors, including those of interleukin -2, -4, -7, and -21, and is thus referred to as the common gamma chain. Mutation in this gene cause X-linked severe combined immunodeficiency (SCID). JAK linked genus kinase 3 genes. Encoded by this gene is a member of genus kinase JAK, family of tyrosine kinases involved in cytokine receptor-mediated intracellular signal transduction. It is predominantly expressed in immune cells and transduces a signal in response to its activation via tyrosine phosphorylation by interleukin receptors. Mutations in these genes are associated with autosomal SCID. For treatment, first in gene therapy, using a self-inactivation lentiviral vector to transduce autologous CD34 plus hematopoietic stem cells. Stem cells are obtained from the patient's bone marrow. The normal gene is inserted into the stem cells using a carrier known as a modified vector, and the corrected cells are returned to the patient in order to restore children's T-cell function. For the medication, children who have SCID with ADA deficiency have been treated somewhat successfully with enzyme replacement therapy called PEG-ADA. This can be related to chemical and cell signaling that help T-cell function. In order to understand SCID is combined multidisciplinary knowledge. To gain more insight, in the research consists with a number of data from statistics from the graph that compare the T-cell after receiving a gene therapy. SCID is globally distributed and rarely occurred. Most commonly, it only affects only male due to an X-linked recessive condition. The family with SCID history of other family members should get genetic tests before having kids. In developing countries, they lack of specialists, doctors, and resources to treat SCID patients. Thanks for watching.